Yeah. Okay, here we go. Okay. So this is
Oh,
like
yeah.
18 years ago. Some of you were babies. Um, so, but we used to work in this depolarized light scattering a lot, and it was for looking at rod-like things that are optically anisotropic. And so I gave this talk in 2002, and it was in Minneapolis. So we had the Mary Tyler Moore thing going on, stuff like that. So idea of this talk, I don't want to talk really about what we were trying to do too much, but just so you know, we were looking at, I think if I get a pointer here, there, laser pointer. We were looking at, how a rod-like thing, like a cellulose nanocrystal, moves in a matrix of other polymers. Okay, and at that time, not much had been done of that, and a lot had been done of looking at spherical objects, how they move in a matrix of other polymers. And so you can actually back out things like what's the viscosity of the solution by following the motion of these things. But we were interested in the rod-like ones because that's what we do in our lab is rod-like polymers for a long time, like forever. Okay. So this is a slide I just inserted this morning. Can you see the thing rotating?
Yes.
Awesome. And so that's a rod rotating between cross polars. So imagine that the laser's coming in over here on the left, going through the solution that contains the rods. But let's suppose we're looking right into the laser. Well, you'd go blind, okay? Except for the fact that we have polarizers in there. And so the incident light is vertically polarized, and we've put a horizontal polarizer off to the side here. So only depolarized light gets sensed. So if we did that and looked at zero angle, this is what we would see. We would see that rotation. And as the thing, it's kind of hard to do. If the rod was vertical, just imagine for a minute the rod is vertical, the electrons in it will get excited up and down, okay, but they don't actually depolarize any light unless it's the light that's polarized is also up and down. If the rod is totally horizontal, not much scattering occurs because the rod usually want to move up and down the length of the rod. It's easier for them. So it's when the rod is at crossed angles, 45 degrees, that you get some light. And that's what this is supposed to be showing, but I think it's kind of out of phase. So anyway, if you're used to looking at Maltese patterns and spherulites, it's basically the same argument that you get the light at the 45 degrees. So anyway, the point is that the light fluctuates, and we can look at those fluctuations by dynamic light scattering. And from the dynamic light scattering fluctuations, we can figure out how fast it's rotating. Okay? So that's not that has optical anisotropy. So the polarizability, if I get out of this and just zoom this and stop this for a minute. Okay, see the polarizability is the term alpha. So that it has to, and that's how easy it is for the electrons to move. And so the electrons have to be more movable in one way than they are in another in order for this to really work. It's really not the rod-like shape, it's whether the electrons move. So we have other particles that are almost spherical, but inside of them they have an optical axis that makes it easy for the molecules to move one way or other. All right. So anyway, does any of that make sense so far? Any questions about that?
What does optical anisotropy mean? Because how would a rod geometry be not optically anisotropic?
What if it's made of if we took okay, so the rod is much bigger than the electron motion, and so so the electrons just barely wiggle, like half an angstrom probably.
Mm-hmm.
And so you could imagine if I took a bunch of objects that were made of tiny spheres, like 100 nanometer spheres, and I just pack them into the shape of a rod, and I say, okay, so the electrons inside those tiny spheres are just sort of moving up and down, you know, a little bit. It doesn't matter to them that they're in this rod. But if the
Right.
things, if the bonds in the molecules are intrinsically aligned, you know, like if it's a backbone conjugated polymer or something like that, then
Right.
there's really a preferential motion at the atomic scale.
Yeah.
So it's kind of optically anisotropic, means that at the atomic scale, it's easier for the electrons to move in one direction than it is in another. And it's all depends on the chemistry, really. And whether crystals, many crystals are.
So within the crystal structure within the crystal, like
Yeah, you could imagine it. Inside the crystal,
yeah,
there's a
okay.
sort of an asymmetry there.
Right, right.
So if you do cubic crystals, I don't think this will work. But if you did some orthorhombics, maybe they might work.
Right.
So actually embedding crystals into these particles, if you embedded a crystal into a spherical particle, it should give a pretty strong signal for this.
And so why did you
Okay.
say the rod is the rod really rotating like that?
Yeah, it really rotates by Brownian random motion. Normally we
So
think of the molecules just sort of wiggling, you know,
right, but
but
is
they
it in a
also,
dilute? You're talking about a
yeah,
very dilute solution?
it's dilute solutions.
Okay.
Yeah, except that it has all these other polymers present to slow it down.
Right, right.
Okay, so go back to this other mode. So, you know, here's dynamic light scattering. The normal one that we do is vertical polarized input and side beam. At angle theta, we take a PMT and we look at the scattered light. And that's called a UV experiment. Does not mean it means unpolarized detection of vertically polarized incident light. So here the light is vertically polarized. The detector is really in an XY plane here, you know, and so we have no analyzer, no polarizing element on the detector in this upper figure. So that's unpolarized detection of vertically polarized incident light. And then this is horizontally polarized detection. Here we put in a horizontal polarizer. The only light that can reach the detector now has to be taken by the molecule and grabbed and shaken in a different direction. So when the light hits the molecule, I don't know if you got me on the camera or not, but if you do, I'm wiggling my hands, okay? So if the light is coming in an up and down motion, the molecule is taking it sort of sideways. And when you take them sideways, it gives us that horizontal component. We get that depolarized signal. And that lets us actually measure the rotational diffusion coefficient, in addition to the usual translational diffusion coefficient that we get. Okay, so I don't want to get that. I don't want to do that. We were studying tobacco virus. I guess I can show you tobacco virus being grown. We actually used to have our own little farm at LSU. We had a farm at one of the LSU greenhouses, and you can see this plant here is infected actually with the virus. That's why the leaves are all crinkly like that. And so we take all those leaves and ground them up and centrifuge them out and do all sorts of stuff to them and isolate, and you get particles that look like this. So Zen, you would love to have these particles there.
Yeah, how consistent they are. They look just so
Sweet.
perfect geometry.
They are wonderful. It's nature made. The only problem is that they break rather easily, but we can get you lots of images of these. You know, that might be a good thing for your system to test.
Yeah, to test it.
It's kind of the same size lengthwise, similar width, a little narrower probably. And we can get you lots of pictures of these in the literature, but, you know, I think we still have some of this stuff. We can get more pictures of it.
Is the virus? This is the virus?
That's a plant virus.
Why is it rod-like? Are viruses
Yeah, they
rod-like?
do that. That plant viruses often are rod-like.
Just
We're
Can,
so used to seeing this coronavirus that's a round virus.
is it because they can get in those trachees, the xylems, and
I
the
don't know.
plant
I don't know.
blood vessels? I don't know. Okay.
I don't know why it is. I think it's just, it's evolved that way.
Water vessels. Yeah.
And we actually have some of this stuff. The way you grow it is you grow the plants as seedlings. Here's some plants being grown as seedlings. You can see how many we grew. And when they get to be 6 inches tall or so, you lightly scratch them with a sandpaper, just barely touch the leaves with some sandpaper. And then you rub on a little bit of a virus that somebody else previously isolated. Or you can take just an already infected leaf if you find it, and just rub it onto that leaf, and pretty soon the whole plant will be growing virus for you. It's really awesome. It
Oh
doesn't
my
kill the plant, but it ruins the crop. It's the tobacco. People who make smoking tobacco do not want this. And if you've never seen tobacco plants, they're beautiful. They're just beautiful.
Was this funded by a cigarette company or
No,
a
this was
tobacco
funded by
company?
NSF. They used to have real science at NSF. I think those days are gone. So
Yeah.
anyway, here's the translation in black and the rotation in blue as a function of concentration of the virus. Okay,
Very cool.
and there basically isn't much concentration dependence to either one. The virus rotates at about 300 cycles per second. That's about how fast it's rotating. And that really should be radians per second, I guess, 300 radians per second, whatever that means. And this is what the actual signals look like in the correlation function. I think probably only Luke has done this experiment a little bit in the labs. But anyway, the point is that we've added this other stuff. Dextran polymers have been added to try to slow the virus down, and we're questioning, how much does it slow down and stuff like that. That's the kind of thing we were studying. And the point is that the virus way outscatters the dextran matrix, even though the dextran is pretty concentrated. I got a lot more dextran here, 28% dextran, and it's almost scattering nothing. That's the green curve here. That's almost no scattering. And the reason for that is because it's between cross polars. The dextran doesn't have any depolarization. So I'm thinking that the CNCs probably do have a depolarization to them. I'll bet it's much stronger than TMB. In fact, I know a group already studied this. Okay, so there's a group that's already done the basic experiment. So anyway, so this is just showing you what you expect out of those equations that Luke would know. Gamma versus Q squared is a nice straight linear plot with slope that gives you the translational diffusion coefficient. And then the depolarized signal is that same slope, almost the same slope, but with this finite intercept. And that intercept, if you go back and look at those equations, that intercept is 6 times the rotational diffusion coefficient. So if I divide this number, about 1800 or 1900, divide that by 6, I got my 320 rotational diffusion coefficient for that. But when I add the lots of dextran, both things slow down. The slope goes down, so it's translating slower, and the intercept goes down, so it's rotating slower. So basically, this experiment works. So this is a quote from Röntgen, the guy who invented X-ray, discovered X-rays. I didn't think, I experimented. So that's what we decided to do. We didn't think, we just started doing these experiments. It's so cool. We were seeing the rotation go down as we add the dextran. We're seeing weight percent dextran going down. I did these experiments myself, most of these. I just took one summer off, and that's what I did. It was so fun.
Question.
And then we wrote that paper really quick. We had some Turkish collaborators. This is with Turkish collaborators, Sezen, from METU, Zuhal Küçükyüz and Savas Küçükyüz. And they came over to Baton Rouge. They thought they were going to die because of the humidity, but they made it through. So
This is the question.
They were from which city?
They're from METU in
Ankara, the capital.
Yeah, from Ankara. That's right.
Mm-hmm.
Yeah.
Yeah, I would imagine.
Have you visited that campus?
I was born in Ankara, and that campus was one of my second options to my real university. Yeah.
Where did you actually go again?
It's a private university, Koç, KOC.
I know it. Yeah.
It's a small one, but it's a good one.
Yeah.
Where is it?
Istanbul.
Yeah. Istanbul is humid, but it's not humid like Baton Rouge. But Ankara is a desert, you know?
Yeah, Ankara, exactly. Yeah.
Yeah. So they thought they were going to die in the humidity here. I got to tell you, it's really humid here today. It's like going to be 95 and 60 or 70% humidity. It's going to be 95 and foggy. And they never actually seen rain either. That's the other thing. They were just terrified by thunderstorms because they'd never really seen a proper thunderstorm.
Yeah.
They'd been to the U.S. before, but never got thunderstormed. Really sweet, nice people. I hope they're well. They're getting older now. So anyway, you can take the ratio of rotation to translation, and sure enough, rotation seems like it's stopping faster. So we were very excited because this is evidence for entanglement effects, which is a big deal in those days. We were really excited. We write this paper, we send it to Macromolecules, instantly gets published, boom. Okay. I don't want to talk about that. I want to talk about that. And then I had a student come along, Randy Cush, and you see how many more points he did compared to me. Look how many points he did. He did phycol. Now, phycol is like dextran, but it can't entangle because it's heavily cross-linked. Okay, so it just goes, the rotation goes down, the translation goes down, these beautiful exponential decays in, mobility as a function of concentration. Each of these translational diffusion coefficients is like 6 measurements. And so this is a lot of measurements and a lot of analysis. So we were surprised, though, that the rotational diffusion coefficient still was diving so quickly in phycol. We didn't think that made any sense. And so Randy decided maybe it was time to think and not follow Röntgen's advice anymore. We should really think now. And it occurred to us finally, this was his idea. He's a smart student. I have kind of lost track of Randy. He probably became a millionaire. He was on his way to being a millionaire in entrepreneurship. Here's our light beam coming into the sample. Okay. And Sezen, this is what CNCs will do too. They'll do the same thing. When the light comes into the sample, it will be rotated because the sample is optically active. It's basically a sugar, and sugars are optically active molecules, and when light comes through them, it gets rotated. So by the time it gets to the center, it's no longer vertically polarized. Okay? So then the scattering occurs, induced by motions up and down where I'm rubbing now. I'm going up and down on this arrow. The scattering is induced by electrons moving in that direction, but see, that produces a polarized signal in a way that you can detect, which you really wouldn't have if the polarizers were working. So it sort of slightly defeats the polarizers. Then the scattered light, after it hits the molecule there at the middle of the cell, it actually has to get out of the cell, so it gets rotated further. As on its way out of the cell, it gets rotated again. So it's kind of a total disaster. And so Randy finally realized that this is what was going on, and he fixed it. And so, so we were getting a little bit of light from the misalignment, with a little bit of the really depolarized light, and we were inadvertently lowering what we thought was the rotational diffusion coefficient because we were leaking through. This other light was leaking through because of the optical active effect. Nobody had thought of this before. Other people have done these measurements and nobody thought of it before. So I feel like Donald Trump. Hey, nobody knew. But anyway, once you fix that effect, what you find is that really the rotation just marches along pretty much normally. But we actually, this talk that I gave at the Society of Rheology, this is the Society of Rheology meeting, was really to repair the fix to claim that one of our own papers was wrong. So I decided at that point that that's a pretty good thing to do. As long as you do it before somebody else does, it's fine. So we actually got 2 papers out of it, the original and then the good one. And then there's some other stuff here that I want to talk about. Well, what I want to do is point out the main thing is, here's our friends from MetU.
Yeah, I'll check them.
Yeah, see if they're still around. I bet they retired. They were so nice. They were the nicest people ever. The point is that, Suzanne is doing an experiment. She's doing a very elegant image analysis of looking at tons and tons of images of rods. And I bet if we just go to, just go up here to search. Images. What are you seeing? Are you seeing, yeah, look at this one here. Can you see that, Suzanne?
I can't see anything right now. It's just blue screen and
Yeah,
Oh,
there's nothing
no,
wrong.
really? I have to reshare. I have to share it differently. Sorry. Now what do you see?
yep, I see beautiful roads.
Yeah, and you see they tend to break. That's the downside to TMV as a model Rodlike polymer is that it breaks a lot. And it has a particular defect at about 2/3 down the rod. It breaks. So you probably have a bimodal distribution here. This guy, you can see where I'm pointing my arrow. That's probably an intact one. And you see a lot of these little sort of stubby ones like that one and that one and that one.
Yeah.
Yeah, but there's a characteristic breaking pattern. But there'll be lots more. Look at this one. Here's some more. I mean, this thing has been studied since the 1930s. And it was like the first thing anybody ever saw in an electron microscope, practically. So, in fact, it was imaged and understood to be, it was imaged before people even knew what viruses were. It was not known at the time of its discovery that it contained an RNA. It was thought to be some aggregate of proteins only. So here's a picture of its structure. It has an RNA in it, and that answers your question, Pratik, about how the alignment, this way, it just helps the RNA to align in this sort of inner helix.
Mm-hmm.
And other people on this campus, GT campus, do study it. I think Zichen Lin has, and maybe Suprik, I don't know. And there's a hole down the middle. You see it's got this hollow part down the middle. It's really a neat molecule. And people have reconstituted it too. You can take and get the RNA out and isolate all those protein units and just mix them together. It will wrap up into that on its own. It's really cool. It's a very neat thing. But anyway, from our point of view, Suzanne's point of view, is maybe it's, it's a set of images that we can get more, and we can look at 1 billion of them in the literature already and use it to kind of train, to kind of get a feeling for how her code is behaving on something that's, I think, a little simpler than the CNCs.
Exactly, yeah. Because
Since it has a
mostly,
very well-defined width, you know.
yep. Yeah, my code certainly will give way more information compared to the CNC images. Because as you said, the widths are almost the same for each object, and the lengths are like there's a maximum length possible. So
Yeah,
they are also limited, like very limited compared to the CNC size distributions.
right. So, and the nice thing about this is if everybody's going to believe your code because everybody agrees to what this thing does. Everybody says, yeah, 3,000 angstroms, that's how long it is. Maybe it's 3,300, I forget, but it's known, and everybody agrees that it's 180 nanometers wide, angstroms, 3,000 angstroms and 180 angstroms wide. But everybody has agreed on that for a billion years. So this would be great. We can do that was a kind of a good test. But what I'm interested in also is now I should be viewing the PowerPoint again. Is that right?
Yep.
Yeah.
Okay, so if we go look at this equation, for things that depolarize light strongly enough, you can just do the zero angle experiment totally on its own. Okay, you can just do it totally on its own. And it turns out that, oops. It turns out that that dr is kt over 8 pi, viscosity of solution, length. It should be radius. For a round particle, it's radius cubed. For rods, I'll just write it as a proportionality. It's 1 over the length cubed, log length over diameter. And actually the diameter doesn't matter. You can take log L minus log D, and log D will be small compared to log L for thin rods. So anyway, the point is that we can get a size with great sensitivity here. If the length doubles, it will drop the rotational diffusion coefficient by a factor of 8. Okay, which is easily seen. So if the length goes up by 20%, that's 1.2 times 1.2 times 1.2, which is what? 1.7, 1.5, something like that. And so it's easy to see that we have a really sensitive way if we can just do the DR thing. So from the standpoint of an instrument that people can use in a biorefinery, this instrument is super simple. It's a cheap laser. It's some polarizers and a cheap detector and a tube that contains the sample. And you would just load in your cellulose nanocrystals and get a size distribution in a matter of a few minutes that would sample, I don't know, billions of CNCs. So if it works at all, it's going to be a nice little tool for biorefineries. And then, you know, maybe we can train it to do all the extra stuff that your image processing is going to do too. So
And
that's
you said that
where
there are already groups in Georgia Tech doing that?
I don't think there's anybody doing that. There's no other labs
Okay.
in the state of Georgia that can do zero angle depolarized scattering, I think.
I see.
There's only a few people in the world that do it. And we're one of them. DLS is one of our things. So I asked my friend here at LSU who makes CNC solutions. He's really good. He's been working with Robert. We have a paper, as you know. He's going to make me some good CNC dispersions. We never get very good dispersions in our lab. And he's going to make some good ones, and we're going to test that. I'll let you know how that turns out.
Okay.
Yeah. So, well, I've covered everything I wanted to cover during our meeting. It's much more better than normal. Uh, don't save changes. Do I want to save the changes? I don't think so. I'll save the changes.